Okay, good morning from South Africa, everybody. Um, People are still drifting in, so we'll give it another minute or so before I hand over to Bernard, who's our speaker today. In the meantime, I'd like to ask that you please keep your microphones muted and your cameras off for the duration of the seminar. And as we get ready to start, maybe you can share in the chat where you're tuning in from. I'm sitting in Johannesburg. Bernard is in Kruger National Park. So please do let us know where you're joining us from today. Then I'd like to remind everyone that the seminar is being recorded. And the reason we do this is to really enable dissemination of the seminars as widely as possible. And so the seminar within a day or so will be available on the Society for Conservation Biology's Vimeo channel. So you can just go onto Vimeo and search for SCB and you'll find the channel. And then all recordings in the series are on there, as well as other SCB recordings for you to have a look at. Okay, so I know that we've got full set-in from New Zealand. I see we've got people from Bonn in Germany, from the west coast of Scotland, quite a few from Pretoria. So I think I'm going to continue with the introduction and we'll just keep on letting people in as they join. So welcome everybody to the 15th seminar for 2021 in the Society for Conservation Biology's Emerging Issues in Conservation seminar series. In this series, we feature articles selected by the editors that are recently accepted in early online view or recently published in the 3 SCB journals. Those, of course, being Conservation Biology, Conservation Letters, and Conservation Science and Practice. Now, my name is Martin Strauss, and I will be hosting today's seminar. I'm a senior lecturer in the Department of Environmental Sciences at the University of South Africa, and I'm also an associate editor of Conservation Science and Practice, and it's in that kind of capacity that I've been organizing the seminar series for this year. So it's my great pleasure to introduce to you today Bernard Coetzee. Bernard is a senior lecturer at the Department of Zoology and Entomology at the University of Pretoria. And for those not familiar with the South African system, that's the equivalent of an assistant professor in the USA. So Bernard's research has a strong focus on climate and land use change and on birds. And he drives this research from his research base in Kruger National Park. He's particularly interested in understanding how humans are altering the natural environment and what this means for our well-being. Now, Bernard is the recipient of the prestigious Jennifer Ward Oppenheimer Research Grant for the period 2021 to 2023, which supports African-led research that addresses issues of concern in Africa. And his Jennifer Ward Oppenheimer funded research program will investigate the impact of light pollution on biodiversity and disease vector ecology. So Bernard is going to speak to us for about half an hour, give or take, whereafter there will be time for questions and discussion. Now the chat is activated. You're welcome to post comments during the presentation and even your questions in the chat. Or if you prefer to ask your questions live, you can, at the end of the seminar, when we open the floor for discussion, you're welcome to unmute yourself and to pose your question to Bernard. And with that, Bernard, over to you.
Thank you. Thank you so much, Martin. I appreciate being here today. Yeah, thank you for the introduction. I'm Bernard, and I'm here to talk to you today about counterfactuals, right? And so I think for some of you it might be completely new, and for some of you it might not be. I'll dissect it in a bit of detail. I was very delighted to be involved in the series. It's been really cool so far. There've been some cracking talks, but at the same time I was also absolutely mortified to give a seminar on this particular paper, right? Because this is a sort of theoretical critique on a rather, in some circles, fringe statistical or technique in conservation science. And so it's not my usual fare. I also must confess that I no longer work very actively in impact evaluation. So a lot of my things now, like Martin said, I'm shifting towards light pollution, for instance. And so it's all been a bit yeah, it's all a bit crazy, but let's power through and let's see how we go. Unfortunately, I'm talking to you from the Kruger National Park today, but this is not like a lion-focused sort of talk, for instance, right? Firstly, just selfishly, some advertisement of I put the post there in the chat box, but please have a look at this Jennifer Ward Oppenheimer Research Grant. They just launched today again, the open call. And so they fund my work, and I'm an externally funded academic, and so yeah, it's a fantastic initiative. It's $150,000 for African research for African researchers, right? And so that please go check that out. And I put my website in there with my Google Scholar links and what the work's about and that kind of stuff if you're interested. Okay, so that puts us off to a start. From the beginning, I need to absolve my co-author, Kevin Gaston. Of course, we wrote the paper together, but these opinions in the talk today really are sort of my own reflections on this work. Okay, so let's get it going. Okay, counterfactuals. It's more of it's coming in a second. I'll talk a lot about this in a while, but it's quite difficult to judge this audience right here today because I don't know exactly what your background is or what your interests are. But so as a very brief introduction, and I'll roll out a few examples later, they are used in a range of conservation impact assessments, right? So trying to understand if we enable a conservation intervention and we get some outcome, how do we really measure what that impact was, right? That's conservation impact. And so it's a family of techniques, right? A range of techniques actually to better establish controls in when you're rolling out experiments. But basically it aims to test the what if scenario. So it's you can almost think of it as a null. So you create a counterfactual and you test your experimental data against that. Okay. And so on this image, I hope you can see my little cursor, but that's like a classical image of deforestation in Brazil somewhere. And let's, for instance, say the green bits are pristine rainforest and the outside bits are now getting eroded for soybean plantations. And so it might seem an obvious comparison to say if you want to test protected area effectiveness, you compare inside the reserve to areas outside the reserve, right? And you get some measure of deforestation risk or changes in bat communities or whatever the case is. But the counterfactual perspective to agree questions that, because we know that there are other correlates that cause protected areas to be placed in a certain place, right? And so actually, are you measuring conservation impact or you're just measuring those covariates or those correlates, right? So in this case, it might be swirl type for whatever reason. Those swirls, I guess that's soybean or whatever the case is. That's where you'd put in your control bits. And so how do we tease apart what these are? And the way to do that, one of the suggested ways to do that are counterfactuals. Okay, and I'll expand. I'll give you a couple of examples to sort of contextualize a lot better this work. Also, this is this like I said, it was a policy piece or a perspective piece, and so they're really short and sort of pithy. I'm going to try and keep the talk like that as well. But please think about questions if you have. I'll definitely be here for the full hour if you want to ask more questions about this kind of stuff. And if I don't work on it, I can send you into at least point you to the people that do this. There's another sort of advertisement. There is something called Team Counterfactual at the Society for Conservation Biology, and so that brings together a lot of people with the required expertise that work on these kind of things that I'm a sort of de facto member of. And so we can really, sort of assist you in trying to do these things better. So you might be asking why? What led us to write this manuscript? And what I'm trying to do here is to transfer lessons that I learned from elsewhere into how I see counterfactuals getting operationalized into conservation science, right? And so that's a tricky place to be. So you might have heard of bioclimatic envelope models, and this really, I'm trying to reflect on how that came into the literature and what this means for how we interpret how counterfactuals are coming into the literature and how people are applying it, right? That's really the basis for this paper and the critique. So just very briefly, this isn't a bioclimatic envelope modeling talk, but basically you fit some surface, like a climate surface, over species distribution records. Some model comes out and you come out with a prediction for probability of occurrence of your species. And there was this wonderful wave in, say, 2004, 2005, when I was a master's student. I just hit this wave very, very nicely, and we could quickly pick up the tools, pick up the analysis. You could run your analysis, you could publish cool papers, you could get a lot of citations, and it was this real wonderful wave of work exploding into the literature, right? But then some people started really delving into the nitty-gritty of this. And so there was a paper, for instance, showing that if you rasterize a classical painting, it wasn't the Mona Lisa, it was some other example, but you can often get better predictions than climate for some of these models, right? And so I thought that was quite a quirky way to go about trying to test them. But it means that sometimes they might not be working the way we think they do, right? And so this led to these big fights in the debate, right? And so I've left the authors out there, but people were saying, you know, if you open the envelope, it doesn't show these climate relationships. And of course, the people who did the climate change work then fought back and said, well, you did it wrong. If you reopen the envelope, it actually makes a big difference. That's something you need to think about. And so that was really interesting to follow. And the point is that I'm not critiquing bioclimatic envelope models. It's still a technique that I use and apply. But it was this progression of how it exploded into the literature, and then people sort of caught up and went, hey, hang on, we need to think carefully about how these methods are really rolling out before we apply them without really, with enough rigor, I guess. And now we're at a stage where, okay, we know the limitations of these models and we try and apply them elsewhere. And so I think the same thing is happening with counterfactuals. And so this paper had a long journey about trying to get into review. It was quite an arduous path. It's yeah, a lot of the ideas are maybe 4 or 5 years old. And so thankfully, a lot of the things I'm raising today, I'm seeing popping up in the literature at other places. And that means people are responding to these sort of thoughts and trying to change the way we use and apply counterfactuals so we optimize the outcomes, right? So we make sure that we're really measuring what we're talking about. If I start talking to people about counterfactuals, I tend to get sort of 4 responses, and I'll run through that because it'll set the scene for what we're talking about. The first response is a what, you know, like sort of the what group. And so people say, you might not have heard of counterfactuals, or you weren't aware that people that do these sort of experiments actually call them counterfactuals. They're quite common in how we use them, but it's sort of a new kid in the block in conservation science. It actually comes from psychology. That's where the literature comes from. I'm not going to talk much about what they are exactly and how they work today. It's quite a complex field, but I've put up some citations there if you want to rather look up at where we sort of stand now and how these things are rolling out in conservation. There's some really cool new work that's come out. The other responses I get from this work are it seems pretty obvious question mark group, right? So people sort of look at these arguments that we've put forward and that I'll present to you today, and sort of don't see what the fuss is about, you know, because it seems like a reasonable way to go about how you work with counterfactuals. And so that's fine. If you're in that camp, then we sort of we're on the same page and we agree with how it works. The problem is that there is a very strong no camp, right? No exclamation. That is really, there's a lot of pushback against trying to understand how these counterfactuals are working and what might be driving some of the biases in how we go about establishing these counterfactuals. And so that was really interesting because these two groups in the middle, that plays out increasingly in the literature, but especially for when this paper was in review. And so it was quite a funny situation where we would get these two various, very different sets of responses. The one response being, oh, that's fine, you know, that's fairly well written, it makes a couple of good points, except, and the other very strong response saying this is completely wrong, you have no idea what you're talking about. And the level of vitriol was quite telling and unnerving. And thankfully that was not in the SCB journals. This is when we were trying to publish this work elsewhere. And so that was telling. I raised this because I think that means we've hit the right nerve. It means that I think we're at the cusp of really thinking carefully about how we use counterfactuals in conservation, and we've got to continue to sort of do so. Okay, hopefully this audience is in the useful category, and that's really where we're pitching this work now. That's aimed at the practitioners. I use that term very loosely. It's not in the classical sense of our practitioners are used in the conservation literature, but it's people who are trying to apply impact evaluation assessments into their work and how this might optimize your work. So if you fall into the 3 categories at the top, you might not find the seminar quite your taste. We're really tailoring it to those practitioners. And like I say, this This is a not a nuts and bolts critique. It's not about the mathematics or the statistics behind this. It's a more sort of broad picture perspective or theoretical idea about how counterfactuals work. Okay, so I hope that sets the scene on why we wrote this paper and why we think it's important. And then hopefully you can where you can use it. Okay, so let's get back to counterfactuals rather than the context for it. This is a more formal definition. It's a bit clunky, but I'll take you through it. So it's a technique or a family of techniques used to establish the difference between the results of a conservation action and that outcome if no action had been taken. Right, that is the counterfactual. So a counterfactual is the outcome had a conservation action or treatment not been applied. Okay, so I'll run you through a few examples if that's new to you, so we can just get on the same page on how that would work. So this is from what I'd call a classical paper by Ana Rodriguez in 2006. And I note that in the paper, she didn't call this counterfactuals, but I think that's what she was doing. It is sort of a new term, right? It's what people were doing anyway, but now it's sort of getting formalized in the way we go about doing impact evaluations. But this graph shows the number of species declines, or the observed number of species declines over time for birds since the 1500s, right? So since records began. And this yellowish portion is the expected to observed decline, and so that's the impact of the Anthropocene, right? Human activities. And now this bluish line is the predicted number of species in the absence of conservation action, right? So the counterfactual in this sense. And that little green portion is impact. That is what conservation has done over the past how many years to try and alleviate that series of decline. Okay, so that's a nice example of how it's been used. But now you can think about how these sort of approaches get applied in all sorts of other contexts, right? And so we won't go through all that literature, but basically you can model what you think would happen to a species in the absence of conservation, right? But the trick here is, of course, that there are different ways to get to that model and get to that answer and set up different counterfactuals. And I'll use that term a lot, counterfactuals with an S at the end. Not just with an L, and the distinction is really important because there's a lot of ways to do this, right? And it's getting applied in all sorts of places. Another way to use this, for instance, is with invasive species. And so if you say, if we didn't apply a conservation action, all these species would just expand, but we applied the removal of the species, and that's impact, right? That's the difference in those two curves. Okay, so that's the species declines curve. That's a bit simpler. Let's get back to sort of that inside-outside protected areas comparison. And so this is from a robust paper that just came out, I hope I'm saying that surname correctly, in Biological Reviews, and they're particularly talking about matching methods, which is a specific way to optimize these sort of approaches. But go check it out. It's a good overview of what's going on here. Okay, and so let me run you through these three panels, and we just talk about the same thing. This green dot is a protected area somewhere in a landscape, right? And the yellow dots are the control areas, the places you would now set up a control to test, is this protected area at a landscape scale reaching the conservation outcome that you want? And that might be the representation and the maintenance of species inside that reserve, right? So this is a common sort of test in this sort of literature. And so you might put a surface at the back here, which is that white to black dots, which is a probability surface, right? And so in this case, it's deforestation probability. Let's say this is in the rainforest somewhere, or slope, because slope influences the chances that a person with a chainsaw can get to a tree and chop it down, right? If it's a steeper slope, they don't have that opportunity. And so the difference there is not because of the projected area, it's because of how the slope works, right? And so what they term in the paper naive, I think that's a bit unfair. They call a naive design is where you just put your dots willy-nilly in the landscape. Okay, and then try and get to an idea of how these sites compare. The other approach would be the buffer zone, that panel in the middle, where you compare just around the protected area to get some sense of what's happening. But they argue you should use a counterfactual approach. And so you can actually only place those control plots inside areas of similar slope to what is in the protected area, right? You can't measure elsewhere in the landscape because then you're not getting a real sense of what that is. And so in this case, it's only slope. but I'll make the bigger point that we don't always know what those covariates are. And the way that this is done then is this panel to the, I'm just moving my boxes here, this panel to the top, where there's a treatment group, and so each dot is an experimental plot or a result that you get out compared to a control group. You can calculate something called a propensity score, and this is basically all those variables or those covariates put together and how they compare between these two different treatment groups, right? And so where they overlap, so we have altitude and elevation and slope and whatever is the same, that's the only place you can really do the counterfactual comparison, because the strength of the answer you get will be more reflective of the conservation intervention, which in this case is a protected area getting established. Okay, so that was a mouthful, but I'm going to use this as an example on where things might be problematic, because there are challenges to applying counterfactuals, and so this is the heart of the manuscript. And we raise these 5 challenges, and like I say, they're sort of broad and conceptual. It's not the nitty-gritty of how this actually gets applied or behind the mathematics. A lot of people are working on that kind of stuff, but it's more trying to think about the broader perspective here. And so the first one, and some of them might seem really obvious, again, like I say, to some of the people who work on this kind of stuff, but that it's not straightforward to applying these counterfactuals or counterfactual thinking consistently. Because we know, and I'll raise this again, that conservation is probably the hardest place to do these sort of sets, because all the covariates can often be correlated with each other. So it's statistically really difficult to get into what's going on into a lot of landscape scale questions, a lot of conservation questions, right? Because there are multiple drivers changing the thing that you're trying to measure. The bigger point here is that they're inherently value laden, right? Because they reflect the estimator's perception of the study system. So in the deforestation example, if you think slope is the thing that is changing deforestation rates, you'll run to slope for its explanatory power, right? And so that's fair. It might reflect, you know, it's not a problem to incorporate your thinking into a study design, but that opens it up for a range of these covariates. And what I don't see people doing is estimating what the influence are of these different covariates when you establishing these broad scale analyses. And so that's the key point. And if this is true, it means that it's opening up a door for intentional misuse, right? And so I don't have any evidence for this. I haven't, like I say, this is a theoretical talk. It's not an evidence-based talk, which is, I'm a numbers person and I usually work only crunching numbers and doing data. And so forgive me on the outset. But if you know what you're doing, you can set counterfactuals to reflect what you think is just happening. And so that's really dangerous in places like biodiversity offsetting, for instance, because a person with nefarious intent can, for instance, show that a company trying to offset its damage it's doing to some landscape is reaching its targets because they can manipulate the counterfactual. So on paper, you can say, oh, we did this correctly because we used counterfactuals in our approach. This is what the literature is recommending is the best approach. But if you aren't explicit about exactly how you put this up and why, how you set those covariates up, then that's lost. And so it's really important to include those into these sorts of assessments for this reason. Okay, that was one. The second one, let me just take another sip, is that great experiments are really difficult in the wicked world of conservation, right? And so owing to the Anthropocene, finding these true controls, especially at a landscape scale, is really, really problematic, right? And I'll offer you a thought experiment. And so remember, this is a thought experiment. I'm not saying this is how it's playing out. That in an extremist case, where there's only protected areas and everything else is destroyed and denuded, you would be unable to use counterfactuals to show that protected areas have a positive effect, right? And so an example would be the protected areas in the Western Cape province of South Africa. And so that's called fynbos. It's a kind of vegetation that occurs there. And it's usually the protected areas on mountainous regions. And the reason they're there is because that's all that's left. That's the only place that you couldn't get a plow or a tractor in to plant wine. So all the places where you could do that in South Africa is wine now. It's not protected at all anymore. And so, of course, that's a bias. It means that those protected areas are places where it's mountainous, okay? But you can't compare that to the fields on the outside because they've been completely destroyed. And so you can't demonstrate to a politician that those places are good to keep around because you can't demonstrate that they're working. And so that's like as in a thought experiment scale, that's a conundrum. So that's not how it's playing out in most places, but it's always good to stretch an idea to its extremes. So, and this also doesn't mean we shouldn't try to apply counterfactuals or think about it. I'm not critiquing the thinking behind this, but I would like to see people be more aware of it, quantify and be transparent about where they think those trade-offs lie. Okay, third in our list of things to think about. Many conservation actions predate the use of explicitly stated counterfactual conditions, right? And so I'll raise this example again of how they demonstrated, I think quite convincingly, that how you can optimize a design based on knowledge of your counterfactuals. But the problem is that most conservation interventions don't work that way. You might have set out for an ecological question, which was, what do the bats do in this landscape versus that landscape, some of which is a protected area. I did some of these analyses. And then you compare inside and outside reserves to get some sense of how it changes over time. But if you don't do it in a counterfactual sense, it throws out that entire analysis, which I think is at risk of ignoring a lot of the way that biologists and ecologists go about doing their business. And so there's a real risk here that we miss what people are doing on the ground just by trying to optimize these statistical techniques. Okay, so these counterfactuals are ideal when used from the outset, but it's really the case. And I think this is really important for things like protected area effectiveness assessments and things like restoration ecology, right? So how do you know where you're compared to some baseline if you create a counterfactual? And you have to be really explicit about creating those counterfactuals, otherwise you're just measuring the wrong thing, right? Or measuring the wrong level of impact or amount of impact. So it's something to think about. Number 4, this is no surprise to people, I hope, in this audience, because we raise this in every SAB meeting and every conference. And it's if anything, this gap's growing, but there's a real big knowing doing gap between research and practitioners, right? And so often people are thinking about and applying statistical techniques and not necessarily considering what practitioners are doing, and vice versa. So there's a lot of practitioners working not being aware of things like counterfactuals and how they can help you and optimize your design and your study design. And so I find that most practitioners are quite a pragmatic bunch, right? They sort of work with that's a compliment. They work with the data that they have available to try and optimize what they're doing. But often that doesn't follow a counterfactual route, because that's sort of a new way. It often follows a strategic adaptive management approach. So you would try and you get some information, you make a decision, you enact your result. It doesn't work the way you thought, and then you try and do it a bit differently, and you sort of do it iteratively until you get to a place where what you're doing is having the desired outcome, right? And so those sort of designs are really difficult to test in a counterfactual sense, of course. And I'm not saying that we shouldn't try to optimize them, but it's a valid way to go about doing conservation. So I get very frustrated with this sort of pushback that the only way to do things, or the only way to set up a real experiment, is to use these counterfactuals, because it's simply not the case. It's not reflective of how people are doing it on the ground. Okay, so this is the last point. The 5th point is sort of a big one, but that a culture of impact evaluation still doesn't really exist in the conservation field. This pops up every now and then in the literature, and this problem is far from trivial, right? Because conservation is probably one of the hardest settings in which to do these impact evaluations, because the covariates are so multiple. There are so many different drivers that could influence biodiversity declining or increasing, for instance, right? It's a really, really complex place to do it. And very few conservationists have familiarity with the mainstream impact evaluation approaches. So these techniques that are getting developed, I find, don't really trickle down to people on the ground. And I think I'm lucky that I'm in a fortunate position where I work next door, in the offices next door to me, to the practitioners, right? People really trying to make a difference on the ground, you know. And so that I guess has changed my perspective on how these things rolled out. I don't see, many of you have done these before, but I don't see grant agencies really providing or ring fencing funding for monitoring and evaluation studies. And usually when they do it, it's sort of piecemeal. It's like, you know, it's like a checkbox, and you say, oh, I reached this, and that, and everyone's happy, and so many smiley faces, and that's it. So that's not a robust way to go about whether your intervention is really reaching the outcomes that you want, which is important, and what's the best way to measure it. And so I think we're missing the boat here a little. And then at the back end of this, maybe a more controversial point is that we're living in this increasing culture of lack of thinking about scientific evidence. And a lot of people, I use that in quotation marks, ignore the evidence anyway. And so I think there's a risk of trying to optimize these approaches, but not sort of missing talking to the audience in the first place, that they're not getting why we're trying to do this, or why it should be important. Now, all of this doesn't mean I'm against using counterfactuals. I just think that we have to ask some questions about how we approach them and how we're using and how we roll it out into conservation assessments. Okay, so what can be done about it? Like I say, I don't actively work in this field anymore. And initially, in some versions of the manuscript, we had promoted a framework that we thought would achieve this. So a way of how we can use these critiques and turn them into positives of working with counterfactuals. I'll hint at it at the bottom, but I'm not going to develop this further. If somebody wants to grab the button and run with this and do this work, you're more than welcome. Okay, so what we're appealing for is more rigor, right? And so what does that mean? It means that we need to be transparent when we report on counterfactuals, how we set them up, what the motivation behind them, some other things that we think could have driven changes in that system that we couldn't account for. Okay, all that information is really important because it helps someone that wasn't there understand why you think your conservation intervention is having that desired effect, whether it be the success or failure of the translocation of a species, whether it be eroding invasive species from a place, whether it's in a restoration ecology context, whether it's a biodiversity offset. You really need to document your thinking behind the decisions you make when you set up these analyses and set up your experiments. Sounds like a simple point, but it's just not getting done. Other approaches are to use multiple counterfactuals, right? Like counterfactuals with an S again. So there are many different ways to set them up, especially if you're using something like a population viability analysis and setting up populations. There's different techniques to do this, and they have pros and cons. And so the way is now to weigh them against each other. And so there's some really cool work that's just come out that I love seeing, because this is exactly what we mean. It's like, hey, man, we've got to think about how this stuff plans out in the literature and how we're using it. So that's lovely. Got to close the knowing doing gap. I'm not sure what the best route for that is, but I like to see initiatives like the SCB now has with this team counterfactual, which is drawn in a lot of the practitioners that often come to those sort of meetings, because that's the people who really need to apply this stuff. It's not just the academic exercise. It has to make difference on the ground for people really making the decisions. I do think there is a lot more need for knowing where the biases lie in using these approaches. I've sort of alluded to a few or named a few. But it's open to interpretation. I'm not quite sure where all of that might play out, but it's a rich place to investigate. Then another solution might be to combine with other approaches, right? So there's different ways to go about how you set up these experiments and why we do so. And a cost-benefit analysis is sort of a low-hanging fruit for me. That was our original framework to try and resolve this, because that's what you make a lot of decisions with, and it's taken the conservation literature by storm, right? The cost benefit return on investment thinking, because it's so useful. You have a set of limited resources and you have to optimize where you spend them so you can get the best return of investment, right? And so you can apply this to counterfactuals as well. You can say, hey, it takes resources to do them. We can only do it that well or this much. What benefit will you get out of that? And is that worth then pursuing? Or should we do it a different way to get more benefit? And so those two fields haven't clashed with each other yet, right? That's still an open question, but I think that there lies a really neat technique or protocol or whatever the case is. Okay, the ultimate sort of take home, I think that what we're on about and what we think should be happening here is that a best practice guidelines must be created for how we apply counterfactuals into conservation impact assessments and conservation thinking. At the moment, people are applying it all over the place, and it's just getting some of it I see really well. It's such convincing analyses. You can't argue with it. It's just done well. Sometimes I look at it and I'm sort of very confused. I've, for instance, reviewed work recently where I couldn't, looking at the supplementary material, where I could demonstrate where they treated different sets of data differently, one as a counterfactual and one not. And that didn't make any sense because they didn't motivate why they were doing it. And so you have to be explicit about this stuff, and so we can understand it. And so one technique I do actively work on is meta-analysis. This is something I really enjoy doing, long-standing interest in that. And so these sort of best practice guidelines exist for meta-analysis. It's not that common in conservation and ecology, but it's increasing. But if you want to publish a meta-analysis in medicine, you can't just submit it in a journal. You can't just do the analysis. You have to go through what's called the Cochrane Collaborations list of or template for how you approach the analysis. And so that means you have to tick certain boxes of ways you approach the data, what you did with the data, which data you included, which data you excluded. It's all transparent. You have to capture it in certain figures. There's some analysis you can do that you report in bias. It's routine now to include those into all meta-analysis. All that stuff just bounces into your supplementary material. But then somebody like me, who might not be in that study system, can look at that data, know exactly what it means, and then I can judge for myself what the strength is of that effect, how strong that conservation impact is. And so that's the key, is to get everyone on the same page so you can judge the information and the science and the data for yourself. And so I guess what I'm really appealing for here is more rigor in how we set up and work with counterfactuals, because we're at risk of misuse, I guess. Okay, I'll leave you with a closing thought. Maybe, hopefully, this stirs up some discussion. This was a quote. The first top one was a quote at a conference, I think at the SAB meeting in Colombia, if any of you were there. But I don't want to say the person's name because I'm scared I misattribute the quote. And so it was sort of meant as a joke, but I think an accurate one of what's going on here. There is no such thing as a counterfactual. There are only counterfactuals with an S in the end. Okay, so that means there are different ways to go about trying to use this technique. If that's the truth, the bias creeps in because different techniques are going to give you different answers. And so it's critical to quantify that so we can make sure that we're doing the right thing. Okay. So that's some food for thought. And the last one is maybe a bit more controversial, is that it's a real false dichotomy to differentiate between counterfactuals or not. I think there's a risk that the field is pushing only these sets of analyses and only these sets of data are useful for impact evaluation assessments. I don't think that's the case. I think there are all sorts of other ways to generate knowledge and information, especially if you're on the ground. So it's really important that we sort of think about these kind of things. Okay. There's a mountain of people to thank. There was a lot of people that helped us author this manuscript and improve it. In the end, thanks so much to the Society for Conservation Biology, Carly Cook and Mark Schwartz and Willem Strauss for putting all this together. I've just joined the CSP team as well as a associate editor, and so I'm looking forward to working with that and expanding that sort of work. So that's nice as well. And thank you to my funders, of course, the Oppenheimer Foundation. And I did put that tag in the chat box. Please have a look at that grant. We look forward to some of those submissions. Okay, and that brings us to the end. I hope there are some questions or some room for discussion, and we can take it from there. Thank you.
Thanks, Bernard. That was great. I was quite glad to see that you made the call there for best practice guidelines development, because I actually had a question, and I was going to ask you whether you feel that conservation science as such needs such a system where we either register proposals of the kind of experiments that we want to do, so that it can go through that review of, let's make sure you tick all the boxes.
Yeah. I mean, I think the I forget the acronym, like Collaboration for Environmental Evidence, that Bill Sutherland runs in the UK. I think that's an attempt to do it, right? So let's collate all the data that's out there on different assessments. It would be interesting to go through it with a counterfactual lens and say how much of that information is actually using which level of counterfactuals. I keep on saying strict counterfactuals versus loose counterfactuals. I don't really know what I'm saying, but neither does anyone else. It's sort of it's like really loosely defined in the literature. No one's really sure what this means. I think there might be some more work to be done in defining exactly what it is we mean with these things. I think in terms of your question, or answering your question directly, it might. I'm not sure who can carry that burden. It sounds like a lot of work. But if you set up those templates, I mean, it's literally an Excel spreadsheet. You say, have you thought about this? Have you reported that? Have you did this? Have you done this? And they're a bit tedious, but it takes you half a morning, and then you know where your analysis stands. it would be tremendously useful as we move forward with this work. So we don't do conservation impact retrospectively the whole time, which is what a lot of people are doing in conservation. We look forward and design the experiments with all these things in place. And then we get more robust answers, so we can do better things on the ground, so we can have better conservation outcomes.
Might be something that journals need to start asking for.
Yes.
That's the best way to drive it.
Yes, I agree, absolutely. And so that, like I say, with the meta-analysis example, some of the journals do, some don't. It's sort of all this. If you send a paper to the Lancet, you won't get it in. They will just reject it. I mean, it doesn't even go through the electronic process. A human doesn't even vet that. It's just, oh, you didn't submit this form. Nope, you can't submit your paper. So that's easy. A computer makes that decision. And journals like the big journals like Science and Nature, now you have to put these assessments with your meta-analysis, otherwise they outright reject the paper. So that, I think you're absolutely correct. The journals can play a big role to do this. Okay.
There's one comment that I'll read now, but in the meantime, if you want to ask Bernard a question straight out, you're welcome to unmute yourself and ask.
Hello, Tony.
Hi. Should I read my comment there?
Tony, you're welcome to. Yeah, you're welcome to.
Yeah, so Bernard, I was just wondering about just going further back and where all these, on this question of conservation effectiveness started, and does it have its roots in academia, with academics just wondering, how many species are we actually protecting, or was it more coming from managers and conservationists who were concerned that a national park might be deproclaimed, or they were trying to justify more funding by showing, actually quantifying some sort of conservation benefit?
Well, that's a cracking question, Tony. That's really good. I mean, I'll give you my perspective, but anyone can chip in here. That's so open-ended that I think a lot of people can answer that. Probably many others are better suited to, or better positioned to do so. I think it's probably a bit of a mix of both, right? So some of it is underpinned by the shifts in the values that underpin why we apply conservation, and so I think people have had a very inward-looking, fortress-based protected areas sort of lens at looking at conservation, and then sort of people went like, but hang on, what's happening in the properties next door, and how is that affecting us? And and so that popped up a lot of places on the ground, at least. And then also on top of that, I guess, is the global scale targets and indicators and initiatives driven by the CBD, for instance. And so the one, and this is what led me down this path, is that the CBD indicator for many years was area of protected areas as a good conservation outcome. That doesn't say anything about ecological integrity or what's actually in those areas, right? It's just a measure of, you can measure it on a map. So there's a lot of reasons to do it, good reasons. You can quantify it on a computer, and it's, governments can do it easily enough, and you can report back on it. But it doesn't tell us much about what's happening on the ground. And I think then this sort of led to the question, but hang on, what are protected areas contributing to the portfolio of conservation? How are they contributing to different aspects of biodiversity conservation? How do we develop indicators for that? And then I wrote for my PhD, I wrote a proposal to do it, to develop the new PA indicators for the CBD. And I was very politely pulled aside by some senior people, and they said, look, this can't be done because the data underpinning it isn't strong enough. And I said, what do you mean? And then I went into all this literature doing these comparisons, and we're just not sure at that stage, like how are these things contributing to the conservation portfolio? And so that sort of led me down this path of how do we now do this, and how do we assess the evidence for these sort of comparisons globally? And then I bumped into the counterfactuals, and that was like a thing you have to wrap your head around as well, in addition to all these other things. So yeah, I'm not sure if that answers you, but probably different sets of places. I do think that it should be done. I think it's common in other fields, like medicine. if you do something in medicine, you have to demonstrate that it's having the desired effect. Otherwise, you might just kill the patient, right? We don't do this in conservation. We don't have that attitude. You have to know if what you're doing is having a desired effect. And so these, this is might be complicated, but we have to resolve these sort of things as we move forward. It's just critical.
Yeah. Thanks.
Cool. I see Phil has a question in the chat box. Maybe I can just go, counterfactuals in some way. I'll read it aloud, just so I can also understand. Are counterfactuals in some way, an intuitive way, we integrate the world? Yes. In the same way that Bayesian inference or adaptive management has been suggested to be. I agree. We have an idea how things work, or what difference to seek evidence to support or refute or refine that understanding. I don't know if that's a question. I think yes. I think counterfactuals are actually quite common. It's like a thought experiment. I mean, a lot of experiments can't happen if you don't first think about counterfactuals. You might not be realizing that that is a formalized term and that there's this whole literature behind it and all this, but what you're actually doing is setting out counterintuitive counterfactuals. I guess a broader comment is that I'm not shooting them down. I'm not saying we shouldn't be thinking about it. I think they have been tremendously useful in other fields, also in ours, to try and develop new hypotheses, to generate new information, to think differently about the world, to make sure that we're thinking, as you point out in your question, how we refute or refine our understanding. I had just as a segue, an anecdote. There's a South Africans like sundowners, which is where you have a beer when the sun goes down. And often you get to meet researchers in our field, right? And so I had a very illuminating conversation with a physicist that works in CERN, using counterfactuals to try and understand some obscure physics problem because they can't create a null for that particular effect. And they use this all the time to try and differentiate this. I can't remember the detail. And I was just like, wow, that's genius. I mean, that's really helpful to think about the world and advance our understanding. For sure.
Paul, you happy with?
If I could just add to that, Bernard.
Sure.
I just wonder whether the fact that it is intuitive explains why you get those various responses, where some people are going, well, yeah, of course, that's the way we think, and other people are sort of wondering how that fits into the paradigm. That you'll always get people coming up and going, well, that's the way we do things. It's just we haven't called it that.
Of course. Yeah, no, I think you're correct. And like I say, that initial response, I think, is changing. Like people are realizing, oh, okay, no, this makes sense. Well, there's this formalized literature, and it's sort of building momentum. So in some ways, these arguments that we laid out in the paper are a bit dated already, because a lot of people are doing this now anyway. It is also encouraging that I get emails, especially from ECRs, saying, this is a verbatim quote, but saying, I thought I was the only person that thought this way. I was like, no, it's okay to question how these approaches roll out. That's fine. That's a reasonable way to go about it. So yeah, I think that's correct.
Well, that's why your paper's very valuable. I think it may feel like it's got outdated, but there'll always be people that will discover that, and it'll be their point of entry into this way of thinking.
Oh, thanks. That's really kind. Yeah, I hope so.
Cool. Then there's a question in the chat from Jennifer Dodd. Apologies if this is a naive question. I'm new to counterfactuals. How do counterfactuals differ from Barkey observations without the before part?
Yes. Okay. That's that's a complicated question to answer quickly. So the Barkey designs can incorporate counterfactuals, and I guess the counterfactual portion comes in with setting up those controls when you're doing the before-after control intervention. So Barkey designs, I think in the paper we call them actually quasi-experimental. Depending on how much control you have over where you set your controls, it changes the strength of the effect that you might get out. And so sometimes, especially for conservation interventions, you don't have control of where your control sites are, right? You're not in control of that. And so that means you get a reduction in the strength of the power of the analysis, at least. And so counterfactuals come in as a way to try and strengthen those controls. And so one way to do that, for instance, is with something called the matching methods. And so I guess to answer your question very briefly, there's some cool papers on this. Maybe start with Rabas, and we make some references in our paper on this as well. But counterfactuals are a way to try and build into your back-key or into your randomized control investigation to try and strengthen what you're getting out of that. So it's like an addition. It's not a new way to do an analysis. It's sort of a way to structure the analysis. Does that make sense? I hope so.
Jennifer, you happy, or you want to follow up?
Yeah, sorry. Yes, thank you. That was, I'm glad it wasn't a completely naive question.
No, that's a great
Thank you.
question, actually. Yeah, it's a really good question. Cool.
Bernard, while people still maybe type in the chat, taking a few steps back to where you listed the usual responses that you get when you mention counterfactuals. I think your third category was the no with an exclamation of we should not be doing this. What are the main reasons for being opposed to the approach?
Well, no, not no in the sense of we shouldn't be doing counterfactuals. No, in the sense of don't question the use of counterfactuals.
Question the use of counterfactuals.
Yeah, so people are like, this is the only way that you can go about evaluating evidence, and I'm like, no, that's not true. And so that's weird. I think a lot of it, you know, it's I'm being a bit facetious there with some of the comments in the reviews, but it's not simply just a point of that we needed to clarify, or that we you know, that the wording was poor or whatever the case is. There's a what I felt at the time when I wrote that, there's a sort of a systematic opposition towards questioning these sort of approaches. I don't think the counterfactuals are just the de facto standard in science. They shouldn't be. They're one of a very useful, but they're part of a toolbox of ways that we need to approach how we do this. And the reason is because practitioners don't adopt them, you know, and so we have to think really carefully about how we do these things, so we make sure that we you know, providing that sort of information that's useful for people. Sorry, I should have clarified that earlier.
No,
Well, you know.
my bad, not yours.
No, okay, cool.
I don't see any other questions in the chat. Are there any more questions that people would like to ask directly? You're welcome to unmute yourself and go.
Just a quick question, Bernard. So maybe in certain areas now we're getting enough data that you can actually look at change over time. So your measure of effectiveness would be, are species persisting in these particular areas, or are they declining as in outside? Do you think that is actually viable anywhere yet in South Africa?
Yeah, in South Africa context, not. I mean, there are sets of data and places you can do that, and actually there are all sorts of ways to do these time for space substitutions. Like I mean, the stuff I did in your neighborhood, Tony, when I was a student up there. But yeah, I think Jonas Geldman and Johanna Eklund are two researchers that are leading the charge there. So they've pulled together these global databases under the World Conservation Monitoring Center in Cambridge. And so they're sitting on a lot of these sort of exact comparisons to try and quantify change over time, because that's ultimately what you want to know, right? Because if you showing a gradient of change between a protected area and not, all you're showing is that there's a difference. You're not showing the rates of decline. And I suspect that this is a sort of a unrelated point, but I suspect the rates of decline in both protected areas and outside landscapes are occurring in biodiversity. It's just that it's slowed down in protected areas, right? Because of extinction effects and allele effects and all that kind of thing. And so that problem, if that's correct, I haven't really seen that being tested well, but certainly it's those analyses are coming. I mean, in the next very short while, globally they're starting to appear. Yeah.
Thanks.
Thank you.
Cool.
Any more questions?
I did have another question.
Please.
You mentioned the is it the SCB counterfactual group?
Yes.
I was wondering if you could just kind of explain how that works, what they do, or
I must say, as somebody that was very fired up and charged up with this stuff a while back, but less so. So I'm not quite on top of it. The person to contact there is Joanna Eklund. She's at the University of Helsinki. I can draw her email address here somewhere. That basically is I think the ECB calls them working groups, and they have to write a proposal and they get vetted through the ECB committee as a working group under the Society for Conservation Biology on particular components. And at that stage, it was basically a sort of meet and greet on people in the society that were interested in these sort of questions, pulling them all together, and then trying to figure out where this might go. They're also doing a session if you're interested in the upcoming meeting in Kigali. So there's a special section that has now been dedicated. It's quite a big session, a workshop or a I don't know, meeting, seminar thing in there with some speakers specifically talking about how we do these impact evaluations in our counterfactuals and that kind of thing come in. So they're picking up steam and pretty active. I think if you really want more up-to-date information, Joanna will be the person to and Louise Glue at WWF in Washington.
Great, thank you.
Sure.
Okay, Bernard, then I see we got one in from Ramya Rupa in the chat.
Okay.
He says, I work as a researcher on countering wildlife trafficking with WCS India. World over, it seems like NGOs use capacity building of enforcement agencies to sort of counter wildlife crime, although there seems to be little to no evidence on its long-term effectiveness. I understand it adds a layer of complexity because of the human subjects. I wonder if you know of folks who work on something like this.
Yeah, that you're hitting the question right on the button there, because these are the kind of questions we need to resolve, right? Like, I mean, how do we use this? And I think that human element is critical to understand, right? And how that works. Off the top of my head, particularly with wildlife trafficking, I can't think of somebody working in the impact evaluation space. The person who might be able to put you into that world is Diego Verissimo. He's based probably in the UK now. I think he's Portuguese, but he works a lot on the sort of the social aspects of conservation biology. So he would know that. He knows that network really well. I don't know that space well. That's one person. I don't know how to spell that, but he's at the WCMC. That's one that comes to mind. Sure, I'll give it a bit of thought. I can get back to you later there if you can tweet me or email me or whatever, and I can see if I can draw some more people together. But that's a really key question, right? Really, really important.
Cool. Yeah, I was going to say perhaps you can, perhaps an email can be sent to you because your email address is on the paper. Just to follow up on that.
Absolutely, very more than welcome to.
Bernard, I was wondering, moving forward with your work, particularly the light and the vector and disease work that you sort of getting off the ground now, are you following this approach, counterfactuals?
No, yeah. I mean, it's a bit of a different question, right? So there, a lot of the work is lab based, and so this, it's I guess in a epistemological sense we're using counterfactuals, but I mean we're setting up before after control intervention, choice experiments, and that kind of thing. So that doesn't quite capture a counterfactual approach. I think for some of the other work that we're doing is sort of wilderness concepts. I even haven't started to think about how you set up a counterfactual for wilderness. Like, you know, I mean, that's like a thought experiment I can't wrap my head around, and so I've been avoiding it on a daily basis. If that answers the question. Thanks, Michael. All the best. And so, yeah, I think it's important. Look, if you can do, I guess the point is, especially at landscape scales, if you can incorporate counterfactual thinking in the onset of the experiment, then this stuff is just going to put you light years ahead of other designs. It's really, really useful. The problem is if we hindcast, right, and try and use existing data sets and sort of cobble together analyses with it, that this really becomes problematic how we apply them.
Yeah. Okay. Thanks, Bernard. That brings us pretty much to the end of our time slot. And I know everybody has got other things to get to. So Bernard, thank you very much for an interesting presentation. And thanks also to all of the attendees for your interaction with Bernard and the good questions and discussion that we got out of you. I'd just like to remind you that next week, same day, at 5 GMT, we will have 2 presentations in the same time slot. Some of you may have seen the special issue on intended consequences that came out in Conservation Science and Practice recently. So we're going to have 2 speakers. One is Claire Hirashiki from the University of California, and then Ben Novak and Michelle Weaver from Revive and Restore.
Oh, cool.
And so Michelle will introduce the 2 speakers, and she was the editor of the special issue. And then the 2 papers that will be covered is concern over hybridization risks should not preclude conservation interventions. And the one by Ben Novak then is US conservation translocations over a century of intended consequences. So I look forward to seeing you again next week, Wednesday at 5 GMT. Thank you very much and goodbye.
Thank you, everyone. Take care.